Thanks for the introduction, and thank you all for staying here and giving me the opportunity to talk about Peter Bio. If I can, yeah, it's going. So we are based in Boston. We started the company in 2018, and what we have set out to do is to become a leading player in the genetic medicine field. And our contribution is that we have been working on a new gene editing platform that's especially focused on in vivo gene editing. In the coming slides, I will give you more background on how the technology works, what's different to other state-of-the-art technologies, and especially why those differences matter. What can you do with it? How can you create different therapies using this platform? A few words on the IP situation. We developed the technology in-house, therefore we don't have any in-licensing. We've also done extensive FTO analysis, so you get a far less convoluted technology when comparing it to some of the other platforms. We have a number of initial targets that we're working on ourselves, but since this is a platform, we can also discuss your targets of interest and how we can work together. So we're privately funded, and we will be raising capital in the second half of this year. So compared to a number of the standard technologies like base editing or prime editing, what's similar is that we can do similar types of editing. So we can do single nucleotide changes, but we can also do more complex edits. Where we are different is in versatility and in safety. So the first big difference is that we've created the humanized editor. And we've done that to avoid immune reactions. And we think that the consequence of that is that you have the potential to do more than one treatment with the same individual. And we think especially for big organs or difficult to reach cell types, that could be quite a significant benefit. We have a very compact size. The constructs are about 300 to 400 amino acids. So when you think about delivery, smaller size makes it a lot easier. And then we also have a protein delivery option. And we think that that can also be quite significantly different because it gives you the potential to reach much deeper into the body. When you compare to, for example, monoclonal antibodies, they go to a lot of different places than, for example, where we can take LNPs at the moment. And then in terms of safety, there's a lot of additional benefit coming from using a humanized construct, but we've also done off-target analysis. We've looked at indels, and there also our system has a lot of advantages. So how did we develop this system? How does it look like? In the end, it's a very simple two-component system. We've created something that we call a zinc finger helicase. So it's not a zinc finger nuclease, it's a helicase. So what we are combining is a domain from human helicase, and what the helicase can do, and also in our zinc finger helicases, is the zinc finger can get the construct to the right place in the genome where you want to make an edit. The helicase domain can open up the DNA double strand at that specific location, and that gives then the opportunity for the second component, being a single-stranded oligonucleotide, to bind to the complementary sequence. And you can include the desired edits in this way. And what we are finding is that in many cell types, the cell's own DNA repair processes can incorporate the mismatch. So what does it look like when you use this system? Here we have a comparison with the Cas9. And what we are showing here is a deep sequencing panel. The top panel is with our zinc finger helicases. And what you can see in the green bar is just the edit that we wanted to make. When we compare it to a nuclease, like a Cas9 nuclease, and we do a very similar analysis, what you see in blue is a lot of deletions. And for those of you who work with nucleases, this is probably not a surprising finding, but what we can see is a much cleaner type of edit. The second example where we have used human hepatocytes, again, the top panel in green represents the mutation that we wanted to create. And then in the bottom panel, we have provided all the inserts and deletions in the form of a histogram. And hopefully what you can see is it's a very boring picture. We see 0 meaning no inserts and no deletions. but then the number of reads that we have, where there's a deletion or insertion, is very, very low. And again, we see this pattern. We see something very similar when we look for off-target analysis. And of course, in gene editing, it's all about delivery. And you can have a great gene editing system, but if you cannot get it to the cells where it needs to go, it's still only half a solution. So our system, we're focused especially looking at liquid nanoparticles and also looked at protein-based delivery. So in the liquid nanoparticle, we can, in one and the same particle, include the zinc finger helicase encoded the messenger RNA. We can include the oligonucleotides, and that single liquid nanoparticle is capable of editing hepatocytes. So this is a standard approach, and what we like about it is that, indeed, if you want to do hepatocyte editing, this is a very good system. What's different is that we can also do a protein-based delivery. And what we have found is that the zinc finger helicases have a couple of unique and very useful features. So in the first place, when we produce these zinc finger helicases as protein, what we're finding is that they have cell-penetrating abilities. And when you look at the similarity with, let's say, synthetic cell-penetrating peptides, you see a lot of similarity. There's a lot of arginine, there's some hydrophobic amino acids. So you can actually add the protein to cells and it will be absorbed. In addition, zinc finger proteins typically work in the nucleus. And that's what we're also finding with the zinc finger helicase. By its own properties, it will migrate into the nucleus, so you get very efficient gene editing. Of course, you also need the oligonucleotides, and we have developed technology where we can make one complex. It's not a covalent binding, it's really with a capsule domain, also a human capsule domain. And what we can in this way do is create this particle that we can add to cell culture, and you get very efficient gene editing. What's also interesting is when you compare the diameter, this is only 5 to 6 nanometers, versus AAV or LNP, which are much bigger particles. So when you think about tissue penetration and where do therapeutic proteins go, you can imagine that this is a very different biodistribution that you can get, and therefore also different therapeutic options. So this is to illustrate how the system works with liquid nanoparticles. Like I said, we make one single nanoparticle that includes both components. We mix them one-to-one. We probably can further optimize this whole system. We use standard methodology, precision nanosystems, really standard LNP composition. And when we did intravenous injection into a mouse model in which the hepatocytes have been replaced by human hepatocytes, then harvest the liver and do genetic analysis, we see that even without doing any optimization, we get at a low dose about 10% hepatocytes editing, with a medium dose about 13%. So for those of you who work with hepatocyte editing, this is starting to get into the therapeutic range. Of course, you want to further optimize this, but we think that this really is demonstrating that you can use this system for hepatocyte editing. There's an example where we used the protein complex, and here we are using neurons. These are mouse neurons, and we've selected mouse neurons not only as a therapeutic target, but also it's difficult to transfect cell type. It's like the ultimate post-mitotic non-dividing cell. So we wanted to make sure that our system is also fully compatible with non-replicating cells. What we have done here is, I'm showing you Sanger sequencing. It's an edit where we want to change 5 nucleotides. And I think what you can appreciate is that it's a very clean Sanger profile, and the 5 nucleotides that we do want to change, you can clearly see the edited sequence. So at the moment, we're working on a set of proof of concept studies. We're talking with industry leaders, we're talking with academic leaders, and what we get as feedback is that people are intrigued with the opportunities that this can provide. And of course, we, as a relatively small company, want to be very selective about what we work on, but we also want to be very selective about where we have the most differentiation and where we can add the highest value to therapy development. So the 4 programs that we are running at the moment, on the top is looking in vivo at hepatocytes targeting the liver. We think the benefit that we there bring is the ability to, the safety profile, but also the ability to first correct the liver, but then still have the opportunity to go into other organs. With many metabolic disorders, the liver may be the primary organ of interest, but often either the kidney or eyes, or there can be other organs that still require further treatment. And this gives the opportunity, that the potential that you can go back and also treat those other organs. The second program that we're running, again, is looking at liver, but we also look at other organs. And the idea here is that by using the protein-based delivery approach, we want to see which other organs we can also select. So while many therapies at the moment seem to be focusing on one organ, and there are indeed many diseases where just fixing one organ is solving the phenotype, is correcting the phenotype. But also many diseases where more than one organ is impacted, or more than one tissue type. And for that, we don't have good therapeutic approaches yet. So that's clearly an area of benefit that we're looking at. Also, we think the repeat dosing can be quite an important benefit. And we think about a larger organ, for example, muscle, or tissues that have turnover. And the fact that for the same patient, you have the potential to do more than one treatment, we think will open the avenue to develop new therapies. I showed you the neuron data, and we know that there's many CNS targets that are highly important. And we think also there that getting to sufficient neurons or other types in the CNS, getting sufficient editing, but especially also tissue penetration. So in the mouse, if you get 2 millimeter tissue penetration, you probably cure the mouse. But if you think about 2 millimeter tissue penetration in human, that may be just the beginning. So again, there we think that the different properties can result in different therapies. And then the 4th program is using blood stem cells. Bone marrow is difficult to reach. Blood stem cells typically are very well protected, but you can also mobilize them. And what we want to look at is, can you mobilize stem cells? And while they're in blood circulation, can you do the editing? What we do know is that there's not sufficient blood stem cells going in circulation when you do a mobilization treatment. So you probably have to do multiple rounds for the same patients. But you can imagine that this could become a very efficient in vivo treatment for for diseases where hematopoietic stem cells are underpinning the disease. And of course, after looking where do we find the most benefit, where it can be most differentiated, that can also be stepping stones to think about other therapies, especially in tissues that are difficult to reach. We'll be selecting the targets that we want to work on, but if you are interested in having a conversation, discussion about potential ways to collaborate, at Peter Bio, in the end, we want to help patients. So the more we can work together, the more we can start to make use of each other's capabilities. We will be very open to that. And with that, I want to thank you for the attention and for the opportunity to introduce Peter Bio.